First of all, I'd like to thank the organizers for giving me the opportunity to speak to you here today. And also I wanted to say that I applaud the FDA and EMEA for organizing this important conference with all the key stakeholders, because I think it's really only by collaborating together and sharing our learned, all the knowledge that will make more rapid progress with this important issue. So I'll be speaking about our industry experience with PML, focusing really on the lessons learned from Tysabri. The topics for my presentation are threefold. I will be describing our risk stratification efforts with respect to Tysabri. I'll talk about the evolving case definitions for PML and conclude briefly on benefit-risk communication. So much has been said already about Tysabri. The main thing I want to highlight is really the fact that it's on the market in more than 50 countries. And the reason it's approved is because of its very significant efficacy in the treatment of a very severe and disabling disease. And the other reason it's approved is because of the global risk management programs that we've implemented together with regulators. And these risk management programs really seek to inform prescribers and patients about the risk of PML. They seek to better characterize the incidence of PML and the risk factors for PML, and we're really showing a lot of progress in this respect through risk stratification efforts, which we hope will further minimize the risk of PML. And finally, these risk management programs seek to improve outcomes of PML in terms of survival and functional status. In the past 5 years, since Tysabri's been on the market, more than 80,000 patients have been treated, representing 150,000 person-years of exposure, and more than 35,000 patients have had over 2 years of treatment. Can you help me advance the slide, please? As other speakers have said, as of early July, we have 145 cases of PML. 57 are from the United States, 81 are from the European Economic Area, and 7 are from the rest of the world. 116 of these patients have survived, so we have an 80% survival rate. As was very eloquently described by Dr. Berger in the first session, the survival rates in natalizumab-associated PML are significantly better than in other patient cohorts with PML. For example, compared to the autoimmune setting, in the transplant setting, and even in the HIV setting post-heart, really these 80% survival rates are the best that have been seen so far. We have done detailed analyses trying to characterize what are the factors that predict who is likely to survive compared to who is likely to not survive. And we've identified that patients who survive are more likely to have shorter duration of symptoms to diagnosis. They tend to have more localized disease on brain MRI at presentation. They tend to be younger and have less pre-PML less disability due to their MS prior to the onset of PML. And so we believe that improved survival in natalizumab-treated patients who develop PML is likely due to a combination of factors. We think that earlier diagnosis is probably an important element, and then additionally prompt cessation and reversal of the immunosuppression through active removal of Tysabri is important, as well as aggressive management of complications such as iris. We've also spent a lot of energy trying to better characterize the functional status in patients who do survive PML. These are data that have been presented by Dr. Keller-Stanislauskas as well as Dr. Berger earlier today. What I'd like to highlight is the patients in whom we have at least 6 months of follow-up since PML diagnosis. What you can see here is that 13% of patients have mild disability based on Karnofsky scores. That means these are patients who are able to work and carry on all activities of daily living. 47% of patients have moderate disability, meaning they need some help with activities of daily living. And then 40% of patients, unfortunately, are severely disabled and need help with all activities of daily living. I think that in the patients who are severely disabled, it's important to point out that most of those are on the cusp between moderate and severe disability. The reason we have picked the Karnofsky score to really characterize this is that it's an instrument that I think is very easy for physicians to fill out without consulting their charts, as opposed to perhaps some of the other instruments like EDSS, which are used more commonly in the clinical trial setting rather than the clinical practice setting. So that's why we focused on Karnofsky scores. We've also tried to get more information on Karnofsky scores prior to the onset of PML and are avidly trying to get that Unfortunately, we're not always successful in getting all this information as much as we'd like. So we do highly encourage physicians to continue to collaborate with us in getting this vital information, I think, in terms of characterizing outcomes. We have 3 identified risk factors for PML now: treatment duration, immunosuppressant use prior to receiving Tysabri, and the presence of anti-JCV antibodies, which is now included in the European product labeling for Tysabri and is under review by FDA. This is the incidence of PML by Tysabri treatment duration. As you can see, in the first year, the risk is very low. It's about 1 in 30,000. In the second year, or 13 to 24 infusions, it's 0.5 per thousand. In the third year, or 25 to 36 infusions, it's 1.9 per thousand. And in patients treated in the fourth year, or 37 to 48 infusions, it's 1.3 per thousand. There's been some discussion earlier this morning around the incidence in the US versus Europe. The point estimates are slightly higher in Europe compared to the US, and we don't have a very good explanation for this, but one lead that we do have is that the prior immunosuppressant use in Europe is somewhat higher than it is in the US. So when we look at data from our observational studies, about 24% of patients in Europe report prior immunosuppressant use, compared to 13% in the US. And that may be a possible reason why we see a very slight difference in the point estimates between the 2 territories. In terms of prior immunosuppressant use, so again, we've identified this as a risk factor for PML in Tysabri-treated patients. The way we identified this was that we realized that in patients who do develop PML, they are much more likely to report a prior history of immunosuppressant use. So about 40% of those patients have such a history. In contrast, when you look at the Tysabri-treated population as a whole, based on data we've collected in an observational study, the rate of prior immunosuppressant use is about 20%. So based on this, we estimate that the risk of PML in patients with prior immunosuppressant use is about 3-fold higher compared to patients who do not have prior immunosuppressant use. Finally, we've been doing a lot of work on the anti-JCV antibody assay as a risk factor for PML. And the rationale for this is that JC virus infection is a key factor for the development of PML. We've developed an antibody assay that has 2 steps, a screening test step and then a confirmatory test step. And that's designed to identify patients who've been exposed to the JC virus. The reason for the 2 steps is really to enhance the sensitivity and specificity of the assay to the best of our capability. We've tested this assay now in many MS patients, and we found that the prevalence is consistently about 50 to 60% across many studies. The prevalence does not seem to be affected by Tysabri treatment duration or by prior immunosuppressant use. The proportion of patients who are seropositive increases at a rate of 2 to 3% annually, and the test has a low analytical false negative rate of about 3%. These are data that we've generated across multiple studies in MS patients, and what you can see is that the prevalence of anti-JCV antibodies is consistently in the range of 50 to 60% across multiple studies in these patients. We were also fortunate to have stored serum in 30 of the patients who developed PML while receiving Tysabri. These were stored sera that were collected prior to the onset of PML. And in all 30 of these patients, when we tested them for the presence of anti-JCV antibodies, all 30 were positive. So the observed 30 out of 30, or 100% proportion of cases of PML who are anti-JCV antibody positive is significantly different from the expected proportion that we would see in the general MS population, which is about 50%. So based on this, we were able to provide an estimate of the incidence of PML now based on all 3 risk factors: anti-JCV antibody status, prior immunosuppressant use, as well as treatment duration. And as you can see, based on this algorithm, the risk of PML in patients who are anti-JCV antibody negative is estimated to be less than 0.2 per thousand. In contrast, when you look in the lower right-hand side of this slide, you can see that patients who have all 3 risk factors, i.e. who are anti-JCV antibody positive, do have prior immunosuppressant use, and have more than 2 years of treatment duration, those patients have a higher risk of PML. That's about 8 per thousand. So what you can see, and then patients who have various permutations of these risk factors are in between in terms of risk. And so what you can see is that there's a significant difference in the risk between patients who are anti-JCV antibody positive and anti-JCV antibody negative. These data are now in the EU prescribing guidelines, so they're available to physicians through that mechanism. They're also being incorporated in the treatment initiation and continuation forms, so they'll be available directly to patients in the European Union. So I think this is our attempt, together with the regulators, to try and provide a more quantitative description of the risk of PML based on the presence or absence of these three risk factors. So we continue to evaluate the use of this assay in terms of its clinical utility for the stratification for PML in large studies, and actually it's been tested in more than 30,000 patients to date. We filed these data to both the European and US regulators at the end of December of last year, and in June of this year it has been included in the Tysabri product label in Europe. So we believe we're really entering into a new era of personalized medicine in that this is the very first biomarker that's being used in MS. And we believe that risk stratification, such as this effort, is likely to be useful so that patients can make more individual benefit-risk treatment decisions regarding the use of Tysabri for the treatment of their MS. Now I'm going to turn to case definitions for PML. We've had many years of evaluating reports of PML in patients treated with Tysabri, and we're extremely diligent in evaluating any potential case of PML. We typically talk to the doctors by phone. We often visit the doctors personally to gather the information, and doctors have been tremendously cooperative in providing us with the information that we need on these cases. We have developed a definition for a confirmed case based on collaboration with PML experts, and it's very consistent with the definition that Dr. Menser presented earlier today. And basically, a confirmed case is a case that has clinical and MRI findings consistent with PML, as well as evidence of JCV DNA in the CSF, preferably using an ultrasensitive quantitative PCR assay. The other way to diagnose PML, which is very infrequent these days, is through the use of a brain biopsy, where you may see evidence of JCV based on immunohistochemistry or in situ hybridization. These criteria for confirmed case of PML are also in general agreement with what's in the published literature. This is a paper by Dr. Paula Cinque and Igor Koralnik, where they too, in 2009, presented a classification for a confirmed case of PML that's very similar to what I showed you on the previous slide. So in general, I think there's very broad agreement, I think, on the criteria for confirmed cases of PML. We do face some very unique challenges in evaluating PML reports in Tysabri-treated patients. Because of the high degree of education that we've been providing on PML, prescribers have a very high level of clinical vigilance for PML, and that's extremely appropriate in the setting of Tysabri. And so any patient who has new neurological signs or symptoms really should be evaluated for possible PML. That really should be number one on the differential diagnosis. We also highly encourage the reporting of possible PML cases to Biogen, and many of the reports of PML are early in their diagnostic workup. Cases are increasingly detected early in their clinical course. Some patients are asymptomatic. We have a small number of patients that were diagnosed just on a routine MRI. Many patients have localized disease on MRI, and we believe this early diagnosis is contributing to the improved survival rates that we're seeing in the natalizumab cohort. Some cases may need repeated MRI and CSF over weeks or months to evaluate. So for example, the MRI may be consistent with PML, but the CSF is negative. And in those settings, we encourage physicians to continue to evaluate the case to really decide whether or not it's a confirmed case or whether it can be ruled out. We have been highly encouraging physicians to use an ultrasensitive quantitative PCR assay. Nevertheless, there are a variety of different assays out there. Some are qualitative, some are quantitative and non-ultrasensitive, and they have varying sensitivities and specificities. So in the real world, what we are encountering is that physicians are using a variety of different assays. And then the differentiation between PML and MS can be challenging because they're both neurological demyelinating diseases. And let me give you an example of this. This is an MRI of a patient on Tysabri who the MRI looked a lot like PML, but this patient ended up having an MS relapse. They were JCV negative in the CSF, and they improved with steroids. And so this underscores some of the challenges, I think, in differentiating between MS, or the underlying disease, and PML itself. So we highly support the development of PML assessment criteria using the Brighton collaboration as outlined by Dr. Menser. We believe that there's a general agreement in the community regarding the definition for a confirmed case of PML. We have to acknowledge that there are no established criteria for the definition of a suspect case, and due to the unique issues with Tysabri and MS, it might be difficult to establish universal criteria for suspect cases, and we very much look forward to further collaboration with regulators and PML experts in this evolving area. Finally, let me touch on benefit-risk communication. I think that what I'd like to say is that we do communicate extensively on PML risk. We provide the number of PML cases, the updated incidence calculations, risk factors, and other key information on disease presentation and outcomes on a monthly basis. These are provided to physicians upon request through our medical information websites and medical affairs personnel globally. We also provide this information to patients where it's allowed by local regulations in a more distilled, high-level sort of fashion. We have been doing some surveys with prescribers about how we provide the information. We did a survey last December and then one in June globally, and the feedback from prescribers has been favorable. And what they've told us that even though we're starting to see a bit more detailed incidence calculations now in labeling, they still very much want the much more detailed information that you can provide really outside of labeling through these types of medical affairs channels. So I think this is an area that physicians feel is very important for them to have, and they also currently continue to want the updates on a monthly basis. There are some challenges in the communicating emerging safety information. We are unable to provide, I guess, emerging safety information in a proactive way, meaning to give it to doctors. We can only give it to doctors if they ask for it. We can't just push it out there to doctors indiscriminately, if you will, because in many territories that's viewed as a promotional activity when a company does that. So we have to be very mindful of that, and that's why we provide the information upon request. We hope very much doctors are aware that this resource is available to them and that we are available as a resource to give them much more detailed information than anything that can be provided in product labeling. And finally, and I think Dr. Suvarna mentioned this, I think, very eloquently, it's very important to provide a balanced view on the benefits and the risks. And we talk a great deal about PML risk, and I think that's appropriate, but I think we mustn't lose the sight of the very important benefits of the therapy and how best to communicate that appropriately to patients in particular. And so I think I urge regulators, I think, to add benefit information to educational materials that are part of the risk management plan. And indeed, there is some benefit information there. We just need to be mindful of how we're providing it in a very patient-friendly way. So in summary then, Biogen and Lon have been communicating extensively on PML risk. We believe there's general agreement regarding definitions for confirmed cases of PML, and we look forward to further defining so-called suspect cases of PML. The PML risk in Tysabri patients is increasingly well characterized, I believe, as a result of the risk management efforts and really the collaborative efforts that we've had with regulators and with the physicians in the community. The majority of patients who experience PML with Tysabri are alive with varying disability, ranging from mild up to severe. Survival outcomes appear to be better than observed in other PML cohorts. We've identified 3 risk factors for PML, and the anti-JCV antibody assay we believe is very promising as it is combined with other known PML risk factors to stratify the risk of PML. And so I think this risk stratification approach we hope will allow much more individualized benefit risk treatment decisions for patients with MS. Thank you very much for your attention.